Hey there! Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're diving into a super cool process that makes that possible. Meiosis. And no, it's not the same as mitosis, though they sound almost like twins. So here's the deal. Mitosis makes identical body cells like your skin cells and stomach cells. That's all about growth and repair. But meiosis is where the magic of genetic variety happens. It's how we get sperm and egg cells, which are called gametes. And unlike body cells with 46 chromosomes, these special cells only have 23. Why? Because when a sperm and egg combine, bang! 46 chromosomes making one awesome new human. Before meiosis even kicks off, there's this super important prep stage called interphase. Picture it like getting ready for a big party. The cell grows, replicates its DNA, and gets everything in order. Once that's done, the real fun begins. Meiosis happens in two rounds of division, meiosis 1 and meiosis 2. So you get two PMATs, prophase, metaphase, anaphase, and telophase. First up is meiosis 1. In prophase 1, the chromosomes get cozy with their homologous pairs like matching socks, and something amazing called crossing over happens. They swap bits of genetic info, creating unique combos. That's why siblings can look so different. In metaphase 1, the homologous chromosomes line up at the equator and attach to spindle fibers from opposite poles. During anaphase 1, spindle fibers separate the homologous chromosomes in each tetrad and pull them to opposite poles of the cell. The cell enters telophase 1 with one chromosome from each homologous pair at separate poles. The spindle fibers disappear and the nuclear membrane reforms around the chromosomes. Both cells now enter the next stage, meiosis 2. However, unlike meiosis 1, DNA does not replicate before meiosis 2 begins. Once again, in prophase 2, the nuclear membrane disappears and spindle fibers fan out from the two sets of paired centrioles. During metaphase 2, the chromosomes in each cell line up at the equator and attach to spindle fibers from both poles. During anaphase 2, the sister chromatids of each chromosome separate and move to opposite poles. Once the sister chromatids separate, they are called chromosomes. Finally, during telophase 2, the spindle fibers disappear and nuclear membranes reform and cytokinesis occurs in both cells. Meiosis 2 ends with 4 genetically different haploid daughter cells, each containing only one set of chromosomes. This whole process creates variety. Thanks to independent assortment and crossing over, no two sperm or egg cells are the same. That's why siblings can be so different. They each come from unique combos of sperm and egg. And one last thing, sometimes chromosomes don't separate properly, which scientists call non-disjunction. This can lead to genetic disorders, just one of the many reasons meiosis is so fascinating. And that's meiosis in a nutshell. Stay curious and keep asking questions.